Hello everybody and welcome to OpenHour this week. We're going to be talking about near-infrared photography, NDVI, hopefully a little bit about infogram.org and a number of other things. I'm just going to put this out there right now. If you're currently watching this as a video, we have plenty of space here on the actual talk, and you can get to that by clicking the link that I just posted. So join us online here for a great discussion this hour. If you'd rather just watch, that's fine as well, and there's a spot on the screen where you can submit comments and questions, and we'll get those right here live. Great. Thanks for joining, and we'll get started. So who's going first here?
Well, I guess I can start off. I'll just give a little bit of an introduction to near-infrared and NDVI and some of the things that we can do with it. And then I think Dorn can talk about some applications that he's worked with. He's done some pretty interesting stuff. And then if people are interested, I can come back and talk a little about some software that exists to work with the near-infrared photography, and then entertain questions. Hopefully there's some questions people have. So can people see the spectrum that I have up on my screen now?
Yes.
Or do okay. Yes. Is that all you can see, or do you also see the other screen in the background? Should I do this? Maybe that's better.
We see us as well. There you go. Yeah,
that'll be a little bit cleaner. Okay, this is just
expand it at all?
Excuse me, expand it. Sure.
Can you expand it at all?
How's that?
Great.
All right. So this is just a diagram that many of you have probably seen, and something similar to this that just kind of describes what the electromagnetic spectrum is. And this is always a good way to introduce what near-infrared light is, or near-infrared energy is. In this figure, we're going from short wavelengths. Down here we're 0.3 micrometers, and then going all the way up to the radar range, or the microwave range, which is where radar operates, which goes here. This one goes all the way up to a meter, 100 centimeters. But the part of the spectrum that we're most interested in is this visible part, which is roughly from 0.4 micrometers to 0.7 micrometers. So the blue part of the spectrum would be the shorter wavelengths, the green's in the middle, and the red is the longer part of the visible wavelengths. And as we get even longer wavelengths, we get into the infrared energy. And the first part of the infrared is the near-infrared, and that's what we're going to be talking about almost exclusively today. But just for your information, there's a section called the middle infrared. Some people call it short wavelength or short wavelength infrared. And there's certainly useful applications for that. Some satellites have mid-infrared bands that are useful for a number of reasons. Some of them, monitoring water content and things like that. And then we get into the thermal. So the long wavelength infrared is thermal infrared, and those are the wavelengths that we use for measuring surface temperature and things like that. So some people get it mixed up when they hear about infrared, that we're detecting infrared. They think it's thermal infrared, and we're actually not in the thermal range. We're in what they call the near infrared, which is not going to detect thermal energy. So let me figure out what I'm going to do next. I'm going to get rid of this one and show another graphic, which shows, I think it's this one. Yeah, this is, here's a graphic that has wavelengths from 0.4 micrometers. This is the blue wavelength. And then, 0.6 would be roughly the beginning of the red. So the near-infrared, if you can see my cursor, it's right where this line, these blue and the green line, look almost vertical. And then it goes over into the near-infrared in this area, and then over to the right-hand side of the plot, it's the middle infrared. And if you look at the blue line, this is what we call a spectral plot. And this is actually a plot from a satellite image that has 220 bands. So it's actually 220 samples along the spectrum. And what we're measuring is percent reflectance. So on the y-axis here, it's measured in percent reflectance from 0% up to 80% up at the top here. And this blue line is what you normally expect a vegetation, like a blade of grass. Green vegetation will have this sort of spectral plot where it has very low visible reflectance and very high near-infrared reflectance. And I'll just point out there's this little bump that you see here in the blue part of the spectrum, or the blue line. This is in the green range, and this is why plants look green to us. So we get humans get the sense that plants are reflecting a lot of green energy. A lot of that reflectance in the green band must be very, very high. And as you see, it's not that high. It's about 15%. And that's, it's usually between 10%, 15%. So the actual reflected green energy is quite low overall. But it looks very, very high to us because our eyes are only sensitive to the visible wavelengths of light. So it reflects a lot more green than it does blue or red. So that's why plants look red. But it's also why we have this perception that plants reflect a lot of green light, and they really don't. So anyway, here we go to the near infrared, and you see this very sharp or very steep line, and this is kind of the principle that NDVI uses, or any of the vegetation indices use, to highlight vegetation over other material. So vegetation has a characteristic, this characteristic curve where it's very high near-infrared reflectivity and very low visible reflectivity. And the standard bands that are used for calculating NDVI would be the red band. So that's this area here. These little red dots are from satellite bands. It's a common, the Landsat satellite. Many of you have probably heard of that. So this is the red Landsat band. This is the near-infrared Landsat band. So by using differences, either subtracting them or doing ratios, you get a good sense for where vegetation is compared to other features that might be on the ground. So I think that's all I have for here. I'm going to look at another plot. Where is it? This one.
Real quick, while you're pulling that up, I'm going to send a reminder out that if anybody wants to join us on the video chat, they can click the link that I've posted and join us here on online live. And if you're interested in just watching, as I see a number of you are right now, you can also post a question on the Q&A, and we'll get that right here live. So just a reminder if you're just joining us. All right, thanks, Ned. Sorry.
Yeah, that's all right. And if people have questions, I don't know how I'll get the questions, but if people can speak up or type in questions as I'm talking. I actually prefer responding to questions and just kind of talking on like this.
Sure, we'll see if anybody types in questions. We'll get them in the sidebar. So okay. I'll bring them to your attention if you don't see them. Super, thank you. So here I just have some plots, some spectral plots for 4 different types of materials. So one is this one in the upper left is for dry grass, and so here we can see we're going from 0.4. Again, that's the blue part of the spectrum. Then 0.5 is green. 0.6 would be red. Then 0.7 we're getting into the near infrared, and it's almost a linear line. I mean, it's almost a straight line. It has a bit of a curve to it. And here we're looking at green grass. So this, again, is a fairly typical vegetation curve. It's very low in the blue. There's a little bump in the green. Here it's up to 10% reflectivity. It goes back down in the red. And then a very steep slope for going up to the near-infrared. This is tar paper, so it's very dark. So the reflectance is fairly low, almost the same across the entire spectrum. It's around 5% from the blue all the way to the near-infrared. And here we have a pine board, which is actually, the shape of the curve is very similar to dry grass. It's just that the reflectivity is a lot higher. So for example, once we get in the near infrared, the reflectivity is quite high. It's about 90%, whereas for the dry grass it was, I don't know, 30, 35%. So the shape of the curve is about the same. It's just the scale is a little bit different. So this is, again, just to show that there are some different
every feature that we see in the ground has a different spectral curve. So some people look at spectral curves as kind of like a fingerprint for identifying features, and that's what we're trying to do with a lot of our analysis. And one other thing, I actually don't have a nice graph or photo for this, but one thing I did want to note is that if you just only are looking at the near-infrared band, it really doesn't look very interesting. There's very little contrast because most features reflect a lot of near-infrared light. So vegetation, soil, if you're just looking at the near-infrared band, it's hard to tell what's vegetation and what's soil because they're both reflecting a lot of near-infrared light. So it's not until we can do these differences or ratios that we begin to see we're able to separate vegetation from soil or something, because soil is going to reflect a lot of near-infrared, but it's also going to reflect a lot of visible light. So if you do a difference or a ratio, it's going to have a very low vegetation index value compared to something like vegetation, like green grass, that has a very low visible but very high near-infrared reflectance. So anyway, maybe Dorn can pick up here. Is that all right, Dorn? Talk about some of your applications.
Yeah, that's fine. I was actually going to pull up. Let's see. I was going to pull up one of the images from our place, but from the last IFarm.
Okay. Here. And after, what I can do later, if people are interested, I can talk a little about some of the filters, how they work, and some of the software people are. I don't know if you can talk about Infogram. I don't have a whole lot of experience with that, but I have some other software that I could talk about.
You think? Oh, you're asking if I should do Infogram?
Can you? I'm sure I could do something.
Yeah. Do you want to start? Well, if you start in on it, and then I'll find I'm going to bring up the image from the IFarm event. We can talk a little bit about what we see there.
We do have a question as well from somebody who's watching online, and it says, is there an easy method to calibrate a camera so we can accurately do comparisons over time?
Yeah, that's a good question. That's something I'm actually working on now. I've been posting a few research notes and hope to get another one out this week. And what I'm trying to do is come up with an easy calibration scheme. So we can basically, what I want to be doing is I want to measure reflectance. Unfortunately, the cameras that we're using, they're not designed to be scientific instruments. Actually, in some ways they are. It's just that they add so much other stuff to it that makes it no longer a scientific instrument, really. And so what I'm trying to do is get a normal photograph and convert these pixel values, which are just basically values that are, they're designed to make a pretty picture, something that's appealing to the human eye. And I'm trying to convert those back into a system where we can actually record percent reflectance. And I've been successful, and I can do it now using just a bright target and a dark target. The problem is that I have to do it using what they call the raw format. And I guess I'm going to kind of make a short detour here. Hopefully it's not too confusing. But when the cameras that we're using, they have a sensor in them, and the sensors are actually what I would consider a scientific instrument. They're measuring effectively how many photons are hitting each sensor. So the more photons, if if double the photons hit the sensor, then the value that's recorded by that sensor is doubled. And it's a linear response like that. So if we have 4 times as many photons hitting the sensor, the value is going to be 4 times as large. And that's what we're really looking for. Now, what the camera manufacturers are doing is they put a filter over there. First of all, so it's only capturing light in the visible range because they don't want contamination from the ultraviolet or the near infrared. And then they're getting this basically the photon counts, and they're converting it into different systems, these different what they call color spaces. So they convert it into a certain color space so they can do manipulation. So when we do white balance and things like that, it's done in one color space, and then they have to convert that color space into what they call the RGB color space, because that's what printers use, and that's what the computers that we use to display images and things like that. So it's doing another conversion to make it basically a pretty picture, something that looks appealing to the human eye. And this calibration process that I'm working on is intending to get it back to a raw format. So anyway, there are ways that you can record raw images, and that's what I'm doing. It's just that it's cumbersome, and I found that most people refuse to do that because it takes a good bit of work, and it also adds some constraints. The images are quite large. They can be 20, 30, 40 megabytes rather than just a couple megabytes, because they're recorded in a format like a TIFF format that doesn't lose information. It also makes it so you can't record images very quickly. Maybe it might take 5 seconds for the camera to record a raw image using CHDK. So there are a lot of reasons that make it a bit cumbersome. So I'm trying to figure out a system where people don't have to use RAW, and to be honest, I haven't been terribly successful. It's very difficult to do the calibration without going through RAW, or without having a lot of color samples that I can use to calibrate the camera. So anyway, I'm sorry if I'm not making a whole lot of sense. I'm trying to make some research notes, so I have posted a few research notes. Hopefully they're useful. And if they're not, feel free to contact me. You can just contact me by email or contact me through the research note. I do try to respond to any types of questions that people have. And usually, after we go back and forth a few times, my explanation becomes a little bit more clear. So if people are interested in these sorts of details, I'm happy to help you. Sometimes it takes a few times going back and forth because my explanations aren't always clear the first time around. It's a long answer to a simple question. But the problem is that it is difficult to come up with a simple calibration when we're working with the JPEG images.
Great, thanks. Thank you for that. And thanks for the question. Gustavo, I can see who it was who posted. Thank you. Doran, are you ready?
Sure. Yeah, absolutely. So, so my interest in the Infogram system is looking at indices so that we can look at crop trials and evaluate different species or different growth rates throughout the season. And so I think the ability to sort of fine-tune the filters is going to be really valuable. But I think, and this is where I'm really excited to have Ned in the conversation, is to be able to, we have NDVI as sort of the standard protocol, but now that we have much higher resolution cameras, and we're able to focus for specific vegetation instead of vegetation in general, I think we have an opportunity to sort of fine-tune that process, and that's where I think Infogram is sort of a way to start exploring that. What starts to pop out with what filters and what bandwidths. So, and I think we're just at the very, very early stages of that. But what I can do is share with you a couple images that I've been working with and research plots, and you can see sort of where I'm going with it. It goes beyond just there's a lot that we can see just in the visual green spectrum, and then trying to also add in not just identification but classification of texture and volume, so that we can really lower the cost of doing agricultural research. So here I'm going to share with you a Google Earth image. Is this coming up?
Yes.
Okay. So here we have these are plots that we did at the first. These were balloon images that were taken at the first IFarm event about 3 years ago, and they were then 3D modeled using a structure for motion type process. This was 123D catch. And so you can see the difference in green intensities here. This is vetch, and then you have grass, which is less dark, and then we also have a texture. And we can also see some other species in just the visual spectrum in the yellows and so forth. And you can also see the rows where you have very little growth where they were recently mowed. So with this, we did. I don't have the NDVI handy for this particular plot, but we didn't see a lot different at that point running the standard protocol, and that's where I'm interested in being able to adjust and use different camera setups as well to try and fine-tune what's going to pop. So for each of these, we also have ground data, so we've actually collected the actual, what is actually in every plot from a species mix, so we can compare what's actually there with what we're seeing in the images, in volume and texture and color. And so that's some of the upcoming work that we're going to do shortly. I can also, just for let's see, what else do we have here? Let's see, I think if I screen share, I'm going to have to go to the Infogram site. We can look at a couple others.
So I have another question here from Jack Summers, just while you're pulling that up. If the reflectivity of the near infrared and visible are so different, how do you keep from saturating the near infrared detector without losing the visibility? Is that what it is?
the visible wavelengths.
Thank you.
Yeah. Well, actually, I mean, saturating can be an issue. One of the things that helps us, is the filters that we use to record the near infrared light tend to attenuate the signal, so it's, you often don't have that saturation with the filters in place, because the filters only let certain wavelengths of light through, so there aren't as many photons hitting the sensors. But it is something that you would want to be careful of. So before you started a mission, you'd want to make sure your shutter speeds were appropriate for the lighting conditions that you were working within. But it's another reason why it's not a huge issue with the consumer cameras is because, again, the consumer cameras they kind of balance, once it goes through the processing chain to a JPEG, it really tries to overcome some of those imbalances because it's trying to make an image that's appealing to the human eye, and even it doesn't know that it's looking at the near infrared light. It just thinks it's the near infrared light is going into each of the red, green, and blue sensors, actually. And I have a figure that I can show you how that works if it's of interest.
Great. Let's check out Infogram, and then maybe we can do that.
Yeah, is that showing up right now?
Yeah. Yep.
Okay. So this image here was taken with a small little remote aircraft with a Canon A2200 with the blue filter, and then run through the Infogram. And what you're looking at are two ponds on the farm that we took, I think, May 19th. And I've been particularly interested in aerial imagery of these ponds because they are on a watershed below a field, which has had some surface compaction, and because of that compaction has had some runoff of phosphorus, which has resulted in algae growth in the ponds. And we've been adjusting field management to increase infiltration and so forth. And what the aerial imagery has started to show is that we can quantify that surface color of the algae visually. And so the Infogram here, we're curious to see if it was picking out where we had the algal growth. And there's some other aquatic growth as well. And to some extent, you can see that around the edges here in this one pond, where it's still light blue, and the green is where you clearly don't have any of that growth, and that's what we see roughly in the pond itself. And you can see over here to the right, this is all heavy cattail growth, which shows up again in that blue. And I can see if we can go back and see the original image. it's a little. You've got a lot of sunlight reflection over here, which I think is a little distracting, and some of the growth over here was not getting picked up as well.
Now, did you say you did this with your blue filter on? Is that why it looks like this? Blue?
That's right. Yep.
Okay.
And so and I think I've got another one of the adjacent pond here where you can really see.
So I have a question. If you're on Infogram and you plug in that photo, or just a regular image, would the blue filter on Infogram do the same thing that your blue filter was doing there?
I'm sorry. Yeah, this was a camera that Chris Fasty had set up with, I believe, the blue filter kit.
Okay.
From Public Lab. So let's see if we can get this one opened up. So this was the original image here, and this one actually, surprisingly, there's very little aquatic growth in this pond showing right now, and you can see it again even without the processing. And we'll go through the blue filter colorized, and it makes it pop a little bit along the edge there. But you can really see where you have the gravel roads and the building and the gravel pile over here. So it does help sort of pick out differences that will just pop a little bit more visually, and then would help if you were to then try to process that and quantify the numbers of pixels of particular colors.
Neat.
And this was...
Oh, sorry.
Oh, go ahead.
Oh, I was just wondering if anybody had questions, you can submit them on the Q&A, or if you're on this call, I see there are a number of you who are on the video chat with us. Go for it and just jump in there.
And I'll just mention one thing that showed in one of those images that Dorn had up is one of the nice features with the near-infrared band is that it's very helpful for distinguishing the land and water interface, because water or near-infrared energy is absorbed by water, whereas the shorter wavelengths, especially the like blue wavelengths, tend to go through and then bounce off whatever's in the water, like the ground or whatever is underneath the water. So people will often use near-infrared bands for detecting the land-water interface.
Yeah, and that certainly does. Yeah, especially if you're trying to sort of quantify, or if you're seeing. I thought it was interesting in that last image where you could very clearly see the where the cattails sort of choked out the water, and they yeah. I'm going to bring up the other slides I've got here. Bear with me here. So I wanted to also sort of mention some of the other sort of where we're looking at going with IFarm here, and looking at sort of meshing some of this work in ground truthing with getting on-the-ground sensor networks associated with it. Can you all see that?
Yeah.
Yep.
So this is sort of where what we're looking at, those are some of the same ponds that we have there, and what we're looking at doing is trying to do more adaptive management of multiple systems on the farm, and so looking at aerial imagery for quantification, and then also putting so that, as I mentioned, we had phosphorus runoff. And phosphorus is fairly expensive to monitor, but some of the other so what we're looking at is, are there other indicators that are less expensive to monitor that can be visually checked? And so algal growth is one of the indicators of phosphorus in freshwater systems. And as we know, taking images is much less expensive than some of the sensing equipment that often has disposable or parts. What's the proper term? They wear out. They have limited life. They corrode in the field. And so this is sort of the general system that we're looking at developing for adaptive management. And so we're looking at how these systems all integrate together, where we have, you know, what we're looking at right here is just the one above ground indicator of photosynthesis or general plant health or plant growth, but we're also looking for indicators through imaging of nutrient uptake and water stress, disease, etc., and indicators of root health and growth rate, diversity, the type of structure, and all of this should be identifiable with the imagery, and then also extending some of the same processes down into the soil. So we're looking at some of the things that we can measure with the on-the-ground sensor network, which we can use to actually ground truth some of what we're seeing in the above-ground measurements, are things like soil moisture, conductivity, even some of the soil chemistry can be identified visually. And so, but some of what Ned was talking about is really important as far as developing the calibration of cameras to be able to make that possible. And so one of the things that I've been most excited about on the soils end, and I think it applies as much to the plant health as well, is a simple calibration protocol, which may include color calibration cards that can be included within the shots, either prior to shooting, calibrating the camera before emission, or actually having calibration cards in the target imagery. And then in the case of, there's a paper that I shared with Ned that they're doing that with soil samples and then referencing that to a large database of those soil samples with a color calibration card, and they're able to tell carbon content, organic matter, carbon-nitrogen ratio, even pH, and some of the micronutrients are showing up. So I see this same approach as far as looking at visual, looking at the infogram as we develop libraries of plants and filters and methods for identifying, you know, in the case of plant growth, you're looking at species and some of these other indicators, nutrient uptake, water stress, disease. What are the indicators that are popping with the various filters, and building essentially a library to be able to, and these color calibration cards to make that a more powerful tool. So I just said a whole bunch, so hopefully somebody else can say something.
Neat, thank you. That was great. I think I told you in an email, this is actually one of the technologies the Public Lab does that I probably know the least about. So it's really interesting for me to hear some of this, and especially where you think it could go in the future. And I love the idea of the bank of all these different things and building off the resource. That's really, really neat. Does anybody have questions or comments? Anything, images they'd like to share that they've done? You can join us here if you're just watching by clicking the link I sent out, or you could submit the questions you have by typing in the Q&A button. Does anybody want to have anything?
I just have one other thing I wanted to say about the calibration question someone raised. I think I probably went over off the deep end a little bit, but if the person asking the question was more interested in just how to compare different missions that they were doing themselves with the same camera, that can be done just by taking a photograph of a target, like a gray target or something that's the same target each time, and then making sure the reflectance or the pixel values would be the same between each mission. So if on the one mission it was pixel values were 10% higher or something like that, you could just reduce those pixel values by 10%. So that's an effective calibration technique as long as everything else was the same. So the camera was the same, the white balance was the same, the camera settings, the shutter speed, and all that were the same. Then you can calibrate between missions just using a gray target or some other target. But if you want to compare your measurements with somebody else, then it gets into the more complicated calibration stuff.
So maybe a question on that. I don't know if I'm coming out there, but a question: how much does the sunlight, or how much does the ambient lighting affect if you're doing multiple passes, let's say every month? How much would the ambient lighting affect what you're actually reading?
It has it can have a very strong effect. So if the ambient light is much stronger, and all the other camera settings are exactly the same. So if in one mission it's the ambient light is quite low, and the second mission it's quite high, and all the camera settings are the same, then on the one mission the photos might be underexposed, and the second mission they might be overexposed. So that's the effect it will have. But if you have some sort of calibration target,
right,
you can basically apply an offset to account for some of that.
Okay.
But again, it depends. As long as you're not changing white balance and those other things, you should be able to do that.
So I guess then the other thing is, how big would your calibration target have to be?
It doesn't have to be very large, because you can just pick. I like to average pixels, so you'd want tens of pixels by tens of pixels. So maybe 30 pixel block by 30 pixels or something like that should be plenty. So that could be just a like a like a poker card size card in a picture that was maybe a couple meters off the ground or something like that. If you're flying from the air, so if you've got a kite, then you'd want the target to be big enough to cover at least a 30 by 30 pixel area. I'm just picking 30 by 30 pixels out of the air, really. But you want to have it large enough so it's not being contaminated from light that's not part of the target. So it doesn't have to be a big part of the.
So then the issue comes in, if you're doing monthly passes, to be able to see differential growth or to see something. The challenge comes in really being able to stack all of those as layers in a GIS format of some sort.
Yeah, that can be a challenge, because we have some software that will do image-to-image registration in an automated fashion, but that's assuming that the scene hasn't changed. And if you're doing every month, the scene's going to change a lot, because whatever you're looking at will have grown. It's going to have different shapes, and the leaves will be in different locations and all that stuff. In that case, it probably makes sense to do a lot of averaging. So you're not doing the comparison at the original pixel level, but you're doing kind of averaging. I don't know how many pixels you'd have to experiment, but maybe five, running a filter over to basically smooth out those pixel values, so there isn't quite as much variation from between pixels.
Well, thank you. Thank you. Fascinating. I've actually got to go. I've got something else that's coming up, but really appreciate this. Very interesting.
And if you have questions further down the line, people are posting good research notes on plots on the plot site, and you're welcome to contact me if I can help in any way. I'm happy to give it a try.
Okay. Thank you.
Thanks for joining, Neil.
Okay.
So, and I just put up an image of some of the ag plots as an example of where I would see when we set up lay these out, we would set corner markers with calibration cards right at the top. And ideally, we actually set that up. I was noticing as I started to import these images into the GIS, into ArcMap. I'm not using QGIS yet, but will be. But that had we laid everything out before we even started, it would have been very easy from a processing perspective to import each of those images into the shapefiles directly, and then have the color, that color calibration card in those raster images, which I think would really help a lot with all the post-processing workflow and really accelerate that. And I see that as we have some of these ground-based sensors too, we can automate some of the ground-based collection. So we're both calibrating the visual, and then we're adding soil moisture and temperature and other things at the same point, and it's also serving as corner markers for layout and making it easier to set these grids up in the field to lower the cost of trialing more things here.
So we have another question from. Oh, sorry, go for it.
No, I go.
Yeah.
Another question.
Yeah, we have a question from Jack Summers. It's, do you envision having a bunch of bandpass filters to get specific wavelength ranges?
Specific wavelengths for what? I missed the last word.
Ranges.
Oh, well, I know people are certainly interested in that, and I think it would be fantastic to get a sensor, basically a monochromatic sensor, so a sensor that's only looking at, one has basically one. Instead of a camera that has a red, green, or blue sensor, have a sensor that's just basically have it a camera so all the sensors are the same, and that way if you put a bandpass filter in it, the camera is just looking at that one wavelength. And if you had several of those cameras, and if they were small cameras, you could put them next to each other and have several very narrow band. You have these narrow bandpass filters, and you could emulate different satellite systems then, and different wavelengths that would be tailored to a specific application. So I think that is something people are interested in. Putting a narrow band filter on these commercial, just the off-the-shelf cameras, I don't see that as being as terribly useful. I know some people are experimenting with it, so maybe they'll prove me wrong. But I think that a better approach would be to use a monochromatic camera with a narrow band pass filter in front of it. I think it would be a much cleaner signal. And I mean, there was some discussion on the Plots website of these people in the astronomy community that they actually grind off, or they use a very light abrasive to get those color filters off of the Bayer filter in cameras. So it turns into this monochromatic system. So each sensor is detecting, a very wide wave band of light, and then you put a narrow bandpass filter in front of it, and you've got a pretty neat camera. But that's, I mean, abrading off the color filter off a Bayer filter, or the color filters off of a Bayer filter, that's way beyond me. I think I'd go through hundreds of cameras before I got that one right.
Great, thanks. Thanks for your question, Jack. Doran, did you want to explain this image, or?
So I was just gonna. This is just some of the imagery from aerial imagery from the same aircraft, the little $300 Bixler, that I just wanted to show that we're moving along it. This is exported from, again, a free program, structure for motion through 123D into SketchUp and then back into Google Earth as a method for communicating some of that, and that being able to import plot data directly into Google Earth as well. But I wanted to follow up again on that individual band filter, because I think at some point that is where we're going to be headed, to identify some of the, if we have very particular missions to identify particular species, that we're going to be targeting particular bands. I know we were looking at sort of invasive species, and their bloom time is a certain time of year, and if we can calibrate it specifically to the flower that's blooming, and we can quantify that, that will be a very rapid assessment, which may be fine with we may be able to do it just fine with the consumer grade setup, but it certainly seems desirable to narrow it right down if we have a very particular mission in mind. I mean, and Ned, it seems like something that, you know, over the next few years ought to be. It's not particularly expensive. It's just a technical process of putting together a number of image chips.
Yeah, and they do sell their little video systems that are they're just, have a sensor that's looking in black and white basically, and those are fairly inexpensive. So it's just, I don't know of any decent quality camera that's like, I'm not sure, well, actually I do know some, but they're thousands and thousands of dollars. But I don't know if, I mean, I think if you looked at Edmund Scientific or something, you'd probably find something that might be worth modifying without costing an arm and a leg. But it can certainly be done with the consumer grade cameras. It's just that Bayer filter, since whatever you're recording, you're trying to put it into these red green. You're running it through another set of filters. So even though you put this narrowband filter in front of the Bayer filter, there's still another set of filters that light has to go through in those consumer cameras, and it attenuates more light, so you have to have a stronger signal, and so anyway, it can be done. It's just not as, I think it could be done a little bit better if we could find some decent cameras to hack or sensors to hack.
I just wanted to end with one more thing that I've been looking at this very simple graph here, and that's I don't know if this makes much sense, except that I see this process that we're going through right now is essentially a validation of indicators, and it gets more and more complex as we validate those indicators. So these libraries to try and quantify and understand what are we seeing, and is it really is it accurate? Are we ground truthing? But the way I look at this this approach is that at some point this is going to lead to much lower cost, much more simple ways of evaluating a lot of these environmental indicators. So just as I mentioned earlier, if we can, if we get the spectral library of a particular type of algae, and we can go after that and quantify it, then and that's something that's easily identifiable and correlates to other indicators, and then folks are able to see that without all the fancy equipment. That's really the ideal here, is we get to the point where folks don't necessarily need to snap a shot with the camera and go through Infogram and put it in the library. It's if you see that it's already culturally assimilated. So it's just that's something that I see this as sort of as a way, as we build this database and library of these new indexes, that it's on the way to something more simple, I guess. What do you think, Ned?
Yeah, I agree. I think we're still pretty low on that going up the hill there. But
Yeah,
it has a lot of potential.
So what would you encourage projects in if you could if you could wish more information and data shared on Public Lab, what would you wish those projects to be on?
You want to take a shot at that first, Doran?
Yeah, I mean, I think when we can start to, I think good documentation along with the imagery, and then getting to the calibration cards is going to be really important. And the more associated, sort of the metadata associated with each of the shots, so we can say, you know, we can start to do species identification at a particular time of year in a particular location is going to be really, really helpful.
Oh, go ahead.
No, go for it.
Well, I was just going to add that in addition to just the spectral information that we're getting, what Doran was just mentioning with it, you know, timing the acquisitions is really critical for identifying certain species, and I mean, it's used a lot in invasive species because a lot of the invasive species they're so successful because they've figured out a way, or I guess you shouldn't say they figured out a way. They're you know, able to green up before other things, or they do something. They use timing, so they use the temporal component to be successful and to outcompete other things. So if you understand that temporal component of what you're trying to detect, it's just additional information that can really help you in your mission.
We have another question from Jack Summers. It says, so the individual photodiodes, I'm going to say, did I botch that?
Diodes.
Have color filters on them, and that's a question. Do they have color filters on them, and does he understand that correctly? And I was wondering how those were wavelength specific.
Yeah, I mean, maybe Doran knows more about those. The ones that I'm familiar with, I don't know of any that have filters, but I'm sure you can find some with filters. I mean, the issue with photodiodes is it's just a single sensor, so it's basically like 1 pixel kind of thing. But Doran, do you know more about photodiodes? What filter options and things like that would be?
I don't.
I mean, you could cover it with a filter, I suppose. You would want to calibrate it so because it wouldn't. I mean, the voltage that it's creating would be different. I'd have to put some thought into that. But someone that might, I don't know if Don is on, but he's looked into photodiodes for a number of different applications. I think he knows the photodiode market much better than
Yeah, because I think they're looking at that for the riffle, I think.
Yeah, I know he's got photodiodes, and most of them that I know are very broadband. So it's I don't know what the wavelength range is. It's at least the visible, I would imagine, and it may go out into the near infrared. I'm not sure.
And we have another question here. Can you talk about the target used for calibration? What is a good material for building it, and how big should it be?
Well, again, it has yet to be built, but I think I was thinking something. It would be similar to, you know, a drive-in post that would sit above the crop, so that could be 6 feet tall, or it could be shorter depending on the crop, and I made of a durable material, either a polyethylene or aluminum-backed material. That's and then the color calibration. You know, I think what the sort of the commercial folks use is I think it is kind of a polyethylene material, isn't it, Ned?
They use all sorts of different stuff. I mean, we used to use just tarps, painted tarps. I mean, there are some nice, you know, very expensive panels with Teflon, Halon, different these coatings that are, you know, designed especially for reflectance targets, but those cost, you know, thousands of dollars.
I would see something really simple that you could even bring out your calibration with you, and it's basically a frame that you can slide it in, so that when you run your imagery, you populate your targets.
I have another question here, and this one's for you, Doran, specifically. Where can I find the IFarm slide that you showed that had the drawing of the tree with the roots in the background and talked about things like soil chemistry?
I'll post that to the IFarm. It's not up there yet. And one of the things I've been talking with Jeff about is ways in which we can share postings back and forth, so that my IFarm and public lab research notes can go back and forth. But I'll make sure that gets up there.
That'd be great. All right. Does anybody have any last questions?
I have a question regarding the calibration and the targets. And thanks for the explanation, by the way. I know a lot more now about the whole calibration thing. I believe it's probably a good idea for public lab to maybe develop something like a cheap calibration target that probably can be offered with the infrograms. I don't know, what do you think about that idea?
I think that's a fantastic idea. Actually, that's one of the, maybe not the next note, but I think the one after the one that I'm hopefully going to do this week. I was going to basically just give a summary of my lessons learned and some things that I would like to see, and that's exactly one of them. It would be nice to see public lab produce a calibration target with known reflectance values that people could use to calibrate their cameras. I think that would be, really, really nice. I think, people would use it. I don't know how many people would use it. I have no idea, but I am a tremendous supporter of that idea.
We got some noise there. I was just going to add. That's one of the things that this outfit, the James Hutton Institute, that was doing the soil imagery, did, was they came up with a calibration card that you could fit in your wallet. Now, it's a little different than what we're talking about with aerial imagery, but if you're doing on the ground plant imagery, a wallet-sized calibration card would be just fine. You just have to have it in the field of view as you take the image. And obviously, the larger your field of view, the bigger your target. But I think a lot could be done, and once you have it calibrated, you can scale these targets as well, and calibration cards. So I think that'd be a fantastic contribution to be able to make.
We have one last question here. Is where is that IFarm link? And Doran, if you send it to me, or just post it right here on the OpenHour page for the IFarm link.
Yeah, I'm going to post it right now.
Okay, great. And we have okay, it's on the chat page here. Got it.
Oh, on the chat page. Yep. Oh, yeah, I should. Yeah.
That's okay. I'll get it out on the OpenHour page. There we go. And now it's posted right on the main page there. Great. Well, oh, we have another question. Are we all right to take another question, or are we?
I'm fine. Yeah.
Okay.
Yeah.
For calibration, could we use a paint sample card from a hardware store?
Oh, interesting.
Yeah, I mean, you can use anything as long as it's been characterized. So, I mean, what I'm using for targets, I have a manila folder that I spray painted black, another one I spray painted white, another one I spray painted gray. I have red cardstock, and a piece of tar paper. And what I did, I sent some samples of these to a colleague at University of New Hampshire who had a desktop spectrometer, and she could scan these and give me the spectral curves. So I know the reflectance values of these materials, but they're just common household materials. The thing that I had access to was this colleague who had a desktop spectrometer. So if you had a paint sample and could get it scanned by someone that had a spectrometer, you could characterize that paint sample.
Is this something the public lab spectrometer could help with?
No, unfortunately, the public lab spectrometer is more of a relative, gives a relative spectral curve, so it doesn't really give intensity. It won't. You can't do reflectance, so it doesn't measure the incoming light. So reflectance is basically the reflected light divided by the incoming light, and the public lab spectrometer is just, it's recording reflected light, but it's not giving you reflectance because you don't know the incoming light. You don't know how strong that incoming light is.
Okay. Well, the calibration and paint sample card, David, that would be a great research note. I think you should. I'm encouraging you to post it. So.
All right, thanks. I mean, the point of that was like people could go to the hardware store and all grab the same color code from like a Benjamin Moore. Now, I don't know if the quality control would be such that they would be comparable, but it might just be like some level of benchmark people could use. Yeah,
it probably can't be much worse than, I have the brand names of the spray paint I use and stuff like that, and it would probably vary a little bit, but for the type of work that we're doing, I'm sure it would be good enough. We're working, I don't want to say that, well, I'll just say that what we're trying to do is really, really difficult when you compare it to, satellite imagery and things like that, because we have very few constraints. The conditions are always different, and since we're looking at such detail, instead of looking at a forest, we're looking at individual components of grass. And each, when we look at a grass leaf, we might have several pixels that covers a single blade of grass, and that blade, each pixel is going to be looking at a different angle on that blade of leaf because the blade is slightly curved. So the way the light is reflected off that blade of grass is going to be different for each of those pixels. And we're trying to make some sense out of this very, very, very complex scene with incredible detail. So we have to be a little careful that we're not trying to get too much information, that we don't want to be looking so much at the pixel by pixel basis. We don't want to be looking at the pixel level. We probably have to do a little averaging and things like that because it's just so complex. These images that we have are so much more complex than, say, a satellite image.
Great. Thank you, everybody, for taking the time and joining us tonight on Open Hour. It's been a really good conversation. I've learned a lot. And just do a quick plug for next week. We're going to be doing a talk on spectrometry, which we've touched on a little bit tonight. And Jeff Warren is going to join us and talk a little bit about opportunities with the oil kit and where potentially we can use the spectrometer to identify samples of different things and hopefully potentially oil. And then we're also going to hear from Amy Soka. I'm going to mess up her last name, but Amy from Australia, who's been using the spectrometer to do water samples and run spectroscopy on water from a mine fire in Latrobe. So just a couple of the things that we'll hear from next week, and hope to see some people. And thank you again so much, Ned and Doran and everybody who helped contribute to the conversation here. Thanks a lot.
Yeah, thank you, Steve. Thanks, everybody, for joining.
Thank you, Ned.
Yeah, thanks, Doran.
Okay.
All right, everybody, have a good day, night, wherever time it is for you.
All right.
Thank you.
See ya.
Good night.
Bye.
Bye.